learning. Today, I will be talking about Nierenberg and Mattei and Nierenberg and Letter's approaches to determining the genetic code. To begin, Nierenberg and Mattei, they started out with a sequence of U's plus 20 amino acids, one of which was radioactive. They also had cell-free extract, which is just what's inside of the cell. They took that and they put it in the solution. And then they filtered it. And after they filtered it, if they got a protein that was radioactive, that means the amino acid or a radioactive amino acid must have been incorporated into the protein. And since the amino acid was radioactive, that means it had to be coded for by the codon that they put in there, which was UUU. So this determined that that codon had to code for that amino acid. So they did this for UUU, AAA, and CCC. So the results of this study were that an RNA sequence codes for one amino acid only. So every specific RNA sequence will code for one amino acid. Nierenberg and Letter's experiment started with a trinucleotide, which was just a single codon that was specific, ribosome, 20 tRNA that were all bound to an amino acid, but only one of those amino acids was radioactive. So once they let this react, they filtered out all of the unbound amino acids. And if radioactive or radioactivity was left on the filter paper, they would know that the radioactive amino acid had been coded for by their specific codon. So by doing this, they determined which codon goes with which amino acid. So they were able to determine 61 codons. So to conclude both these experiments, what it all means is that they were able to identify that each amino acid or each codon goes to an amino acid. So one codon will code for a specific amino acid, and that and what some of these codons were and what they coded for. So that's what they accomplished, and thank you.